good afternoon, and good evening. I'm Hui Kai, head of WuXi AppTec Content Division. On behalf of my colleagues around the world, welcome to the first episode of our new webinar series, Collaborations That Transform. With this new series, we hope to tell stories of breakthroughs, many of them accelerated by collaborations beyond boundaries. So, in honor of the World Neurofibromatosis Awareness Month this May, we thought it would be appropriate to kick off this new series by highlighting this group of rare, debilitating genetic disorders. Today's webinar will feature leading clinicians, drug developers, patients, and nonprofit foundations. These stories, including the first FDA approval of a breakthrough therapy for NF1 patients this April, have a lot to tell us about the transforming power of science and collaborations. Our first panel is perspectives from clinicians and patients. Joining us today are three leading physicians on the front lines of NF1, NF2, and schwannomatosis, the three primary types of NF. We also have the distinct honor of welcoming three NF patients. They will bring a critical perspective to our discussion. This panel will be moderated by Luke Timmerman, founder and editor of Timmerman Report. Luke himself has successfully organized multiple fundraisers for cancer research. So without further ado, please join me in welcoming Luke and the speakers.
Thanks so much, Hui, and to the team at WuXi AppTec for hosting today's webinar on neurofibromatosis. I hope we can all learn a little something from the physician scientists and the patients that we have gathered here today. I think that it would be a great place to start with neurofibromatosis 1, NF1. And maybe we could just start with Dr. Korf from UAB. Could you introduce yourself, say a little bit about what NF1 is and how we have traditionally treated it?
Sure. So my name is Bruce Korf. I'm a medical geneticist at University of Alabama at Birmingham. Have had an interest in neurofibromatosis for 35 or so years, I guess. So neurofibromatosis type 1 is a genetic disorder that affects the body in many distinct ways. It is a gradually, slowly, usually progressive condition. It causes tumors to form along nerves anywhere in the body, very often on the skin where they're quite visible. Some of them are fairly small but can be very numerous. Others can be very large and potentially disfiguring. It can also cause problems in the way bones grow and even cause bone fractures or curvature of the spine. It can cause problems with learning and development. And some people also, it can affect vision, even causing blindness sometimes, and potentially even malignant tumors. One of the really great challenges that a person with NF1 faces is uncertainty, because all of these things could happen, just a few of them could happen, and at any given time, you really never know what direction it's going to go. It does tend to be progressive, but how fast it progresses, how severe it is, really varies from person to person. And historically, really the only thing we could do is to wait and watch and try to treat the symptoms. And for the most part, that amounted to surgery, but it was really a very unsatisfactory approach, because the tumors were generally not possible to remove in their entirety. They tend to grow back. There was no way to predict who was going to develop malignancy and who was not. And the treatment for that, when it occurs, is also very difficult. So it is a condition that is hard to live with just by virtue of the manifestations, but even harder to deal with the uncertainty. I think that's a quick nutshell sketch of it.
Now, just real briefly, how many people have this in the US or worldwide, and what kind of prognosis do they tend to have, or life expectancy?
Well, so the frequency of NF1 is about 1 in 3,000, give or take, worldwide. There really do not seem to be well-documented instances where it's more or less prevalent. And this probably reflects the fact that half the time it occurs due to a new genetic alteration or mutation. And these are natural events that are happening pretty much equally across the world. So 1 in 3,000 roughly is a pretty well accepted number. As far as prognosis, again, it's highly variable. If you look across the board at life expectancy, you will see that there is an average reduction of life expectancy. I think one study showed kind of an average life expectancy shifted into the, if I remember correctly, it was like the late 50s or so. But it's highly variable. There are people that live, I have patients in their 70s or 80s with NF1. The life-threatening complications, fortunately, are rare. Mostly it's malignancy, or in some people, blood vessel problems that can, in rare cases, lead to hemorrhage. So there is a slight decrease, but it's really highly variable, and it includes people that live to a ripe old age.
Okay, so Rennie, you've heard all this. Can you tell us your story, how this affected your family, and how you responded?
Sure. Well, my name is Rennie Moss, and myself and my family, we live in Birmingham, Alabama, which ended up being very fortunate for us that Dr. Korf is our geneticist, our clinician for our family, just 5 minutes away from our home. So our son was diagnosed with NF1 when he was 6 years old, when we discovered a plexiform tumor on his neck. It was sizable, and it was life-threatening in that it was affecting his airway. It was surrounding his carotid arteries, and it was growing. And the initial attempt at chemotherapy failed, as was somewhat expected because there was no approved drug to be able to treat the tumor. And so, like Dr. Korf said, it's very, early on, there was nothing you could do other than watch and wait, or potentially surgery. And surgery had failed for Philip. About a year after our son's diagnosis, our daughter, Helen, was also diagnosed at age 4. And then consequently, because both of our children were diagnosed, it meant one of us had NF, and come to find out it was my husband. So like Dr. Korf says, it could be a very mild presentation, or it could be a very serious presentation. In the case of my husband, he was age 36 when he found out he had neurofibromatosis type 1. So all 3 of my family members are followed by Dr. Korf because of the unpredictability of NF1. It can feel like you're waiting for the shoe to drop, and it can be very controlling as a mother, and the anxiety and the fear knowing what could come to pass, both with your spouse and your children, which is why it's really important to have a clinician nearby that you can rely on and really be your coach to call the shots, and to know what to worry about and what not to worry about as your children develop.
How many years ago did you get that first diagnosis?
Philip was diagnosed in September 2011. He had just started kindergarten about a week before.
Okay. Now, you mentioned that Dr. Korf is nearby, which is really nice. He's also not just a clinician, but a leading geneticist. Dr. Korf, can you say a little bit about the big learnings from genetics in this particular indication?
Sorry,
for NF1 patients, what have we learned about the genetic role in that disease?
Yeah, well, so, it's been known for a very long time that NF1 is a hereditary condition. It can pass from generation to generation. An affected individual has a 50% chance of transmitting it to any child. So we knew that there was a gene. All the evidence suggested that there was really one gene that accounted for it all. And actually in 1990, the gene was identified. One of the earlier examples of a gene discovery in a human genetic disorder. And it became pretty clear pretty quickly just by looking at the structure of the gene, and genes are basically recipes for proteins. And when you looked at the protein structure, it became clear it was a regulator of a cellular signaling pathway that basically controls cell growth. And so that clue came about now about 30 years ago. And over the course of time since then, it's become clear, what the underlying cellular mechanism for tumor formation is, although I don't think we could say that we understand it 100% now. A lot of progress has been made in terms of understanding it. And that's what led to the possibility of developing targeted therapeutics. Meanwhile, it also has made it possible to do genetic testing in affected individuals and members of their family. One of the remarkable things about NF1 is almost no two people have the same exact genetic mutation. There's something like 3,000 different mutations that have been defined. They have in common, the vast majority of them do, that they effectively turn off the gene. And it doesn't seem to matter for most people that much how it turns off the gene. Once it's turned off, it's turned off. So a lot's been learned in 30 years, but it's only recently that that's been possible to translate towards the development of a targeted therapeutic.
Can you speak a little bit to that most recent therapy? Because this is a good news story.
Yeah, so as I mentioned, it was learned that the NF1 gene encodes a protein that controls a cell signaling pathway. You might think of it as the cell has kind of like an antenna that sticks out through the kind of membrane that surrounds the cell, which senses signals in the environment that basically might tell the cell to grow. When the antenna picks up such a signal, it transmits inside the cell a signal which sets up a kind of domino effect. It turns on one protein, which turns on another, which turns on another, and you get this sort of domino effect that eventually hits the cell nucleus, which gives the signal to the cell, let's say it's time to grow or it's time to divide. And so the thinking was, well, if the problem in NF, which is what seems to be the case, is that when you're missing the NF1 gene, what you're really missing is the off switch that causes the cell to say, okay, enough, I've done the growth I'm supposed to do, now let's turn this pathway off. Well, the problem in NF is that pathway gets sort of stuck in the on state once this gene has been altered by mutation. And so the question was, well, okay, if the cell is now activated permanently, can you interfere with that sort of domino effect and block it so that those signals no longer reach the cell nucleus? And there are indeed many dominoes in the pathway, and of course it's not as simple as that. It's not a linear pathway, it's really more of a network. But if you think of it as this, sort of domino effect, the question is, where can you insert a block that prevents the next domino from falling? And a variety of different approaches were tested, and these all are in cell culture systems and in mouse models. And it was found that one particular domino, which goes by the abbreviation of MEK, M-E-K, seems to be a critical one. And when that was inhibited in the mouse models, which developed neurofibromas, it was found that they could actually cause regression of those tumors. And that was the critical clue that set up a human clinical trial with one of these drugs, this particular one being selumetinib, which turned out to be effective not just in the mouse model, but also in the human trial.
So, Rennie, I guess this story was unfolding. You must have been watching it with great interest. How did you get involved? Did anybody in your family, were they a good candidate?
Yes, actually. So after our son's first initial chemotherapy was not successful, we were sent home with nothing. But we did consent to have his medical records sent to the NIH for a promising clinical drug trial, which ended up being the selumetinib trial. And about 3 months later, he was selected to participate in that trial. So this would have been September 2016. We went to the National Institutes of Health and he enrolled. His response has been remarkable. His tumor has shrunk 60% while on the selumetinib drug trial. with relatively low side effects. The first months were very challenging in terms of getting used to the fatigue, a little bit of the nausea, some of the rash, but it really has subsided for him. And so he, at this point, is about to turn 15. And what he would tell you is that nobody asks him what's wrong with his neck anymore, which is really important when you're a teenager, you know, your image. I wish he cared more about his image, but he is almost 15. But truly, that the psychosocial improvement is really what he would say is the outcome. But as parents, we're very aware that what was predicted to come in terms of maybe a tracheotomy or worse with his carotid arteries involved, none of that has come to pass. So significant clinical improvement, and a lot of the risks that we were really worried about are really no longer a concern for us as a family. So it truly is a miracle for our son and for our family to participate in this trial. And we're very excited that now it's FDA approved in the US and is going to be available to others that may benefit as well.
That's a great story. But just to clarify, did he had some kind of like bulging tumor on his neck that was, that he was self-conscious about?
So the reason we found out about it is he was at the beach one summer when he was about 6 years old, and we just noticed, a knot. It looked like a swollen lymph node. We went to his checkup, and his pediatrician gave him a round of antibiotics thinking he just had some kind of infection. And when that didn't reside, they did send him for a surgical biopsy, and the pathology from that did show that it was a plexiform neurofibroma tumor. And at the time, it was very small, but it continued to grow very, very large. So the volume, gosh, Dr. Korf, you'll have to comment for me, but it was growing over 10 to 20% per year. So when your body is growing about 10% a year as a young boy and your tumor is growing faster than that, you can see where the line of concern would cross over the normal growth pattern. So it was threatening his airway in a significant way and other vital structures in his neck. And unlike other parts of the body where maybe surgery can hold something at bay, the neck is sort of the conduit to everything in a young boy or a young person. And so surgery is not an option for him. It would have resulted in a significant loss of life function for him. And it's something we hope we never have to do in terms of surgery. But we understand the risk of potential malignancy, which is why he continues to have regular MRIs and clinician diagnostics each year.
Well, I think it's fair to say that, excuse me, I was going to say that when I first saw him, it was pretty obvious that there was a mass in the neck. And I would assume that most anybody would see it and wonder what it was. Whereas now you have to know what you're looking for to realize that it's there, I think.
Mm-hmm.
That's really a great story, not just in the classic measurable ways that we look at, to see how a drug is responding, but also hearing that, he's essentially got a new outlook on life. Can plan for the future and think about, whatever 14, 15-year-old boys want to do. Okay.
I would love to know what's going on in his mind.
You're his mom. He doesn't want you to know everything.
That's right. It's a different clinical trial altogether.
All right. I think that's a great point to transition to our next story with Dr. Blakely and McKinnon. Dr. Blakely, maybe you can start us off and tell us about NF type 2. What is it, and how have we traditionally treated it?
Yeah, so NF2 is another peripheral nerve sheath tumor condition that is also autosomal dominant in its inheritance pattern and results in many tumors, as does NF1. The difference is what type of tumors and where they are. So these tumors, the ones that are most famous for NF2 are tumors that form on the hearing and balance nerves. Those are called the vestibular nerves, and those tumors are called vestibular schwannomas, which are pretty common tumors in the world, especially in older people over 50, over 60 years old. But in NF2, they happen in young people and they happen on both sides. If you are affecting the nerves that control hearing and balance on both sides, you can imagine that one of the most common complications of NF2 is going deaf early in life, but in a kind of cruel twist, not so early that you go deaf when you're a child and become part of the hearing impaired community, which has a lot of resources and is a very richly and welcoming community. but really when you're in your late 20s or 30s, when most of the people who form your social life and work life are hearing. So that's the most common presentation for most people with NF2. There are many other schwannomas that form. Schwannomas are tumors that form of Schwann cells, which are the cells that line the nerves of the peripheral nervous system. And of course, our peripheral nervous system is everywhere that's not brain and spinal cord. So all of the cranial nerves that make us smile or make our tongue move or make us swallow, and all of the nerves that come out of our spinal cord to make my hand move or make me feel when I scratch my hand. So there can be focal weakness in one arm, one leg, sensory loss in one arm and one leg, or pressure put on the spinal cord that can result in loss of bladder and bowel function, loss of ability to walk. That's all just from schwannomas. And then many people with NF2 also develop meningiomas, which are another tumor that's very common in human beings in general. The challenge in NF2 is their multiplicity. There are so many of them, and they progress slowly over life. And they can cause a lot of pressure on their neighbors, on the brain and spinal cord. And then the last tumor is called an ependymoma. And fortunately, in the setting of NF2, those tend to not require intervention very commonly, but they typically involve the spinal cord. And so if they become symptomatic, they cause spinal cord symptoms.
Like with NF1, what kind of prevalence do you see out there? And again, do you have any data on life expectancy?
Well, there was this really brilliant researcher named Gareth Evans who did a very important study that showed us that it, depending on your mutational subtype in NF2, that predicted to some extent what the prognosis was. And very much like what Dr. Korf was saying, it has to do with how much of the protein is available to the body to use it. So mutations that are truncating, that result in no protein product, have a much more severe prognosis. And then there are a variety of other mutations that have variable prognosis between that. But so that study was really important because it does help us speak with families and patients about prognosis. But I think equally important in that study, it showed that it matters where you get your care, and that getting care in a specialty center where things are done in a tailored and cautious way, playing for the long game to maximize function over decades, really does influence outcome as much. So like NF1, prognosis is a little tricky because there's so many variables that go into it. In terms of prevalence, it's far, far more rare than NF1. It's best estimates at about 1 in 50,000 people have NF2. So it's pretty rare, although I don't feel like it's rare. I think there are a lot of patients who have NF2 and don't know they have it, and their providers don't know they have it until they put the pieces all together and they finally get referred to a specialty center.
Do you often find patients on a diagnostic odyssey before they end up coming to you?
Absolutely. They've almost always started with otolaryngology or neurosurgery, or they were in the ER and told they have metastatic cancer because they see these tumors everywhere, and so they end up in oncology. So I hate that for our patients. I love when I get to say, great news, this is not cancer. A little more complicated news, this is something else that most people haven't heard of, so let's talk it through.
Okay, so McKinnon, you've heard this before. Can you tell us when you got this news of the diagnosis?
Yeah, sure. Hearing all of those things that can go wrong always just kind of makes your anxiety go a little high. But so I got diagnosed about 11 years ago. I was hit in the head with a volleyball, and I went in for an MRI, and they found 2 brain tumors. And I mean, you go to the internet, of course, at first, and it's like, oh no, because there's so many, there's so much information out there. I think, as Rini kind of said, the anxiety is just never ending, which is really, really hard. But I found a specialist. I found Dr. Blakely, and I travel about 9 hours to go see her. By car, by plane, it's a couple. But still, I mean, there needs to be more clinics around the U.S. in general, but I think that over the years there has started to be, which is really exciting. But yeah, so I think just the anxiety in general is something that I battle with.
How, what kind of options were presented to you in terms of treatment or management of the disease?
When I was first diagnosed, or now? Oh, when I was first diagnosed.
Yes.
So when I was first so when I was first diagnosed, a lot of people, even in my area, didn't know what NF is when I would go to doctors and stuff. So I had to kind of educate the doctors on what NF was, which was, a little discouraging. But when I found Dr. Blakely, kind of the options, it was Avastin and surgery and watch and wait and some other trials that hadn't quite come out yet. And that's about it. So it was very limited options, but there were some options.
But surgery, these are tumors in the brain. Were they accessible at all in your case?
Brain surgery was no fun. So I've had 2 brain surgeries. My first one was on my 18th birthday, and I got diagnosed on my 16th birthday. So my first one was on my 18th birthday. My second one when I was 22. And the first one they were able to save my hearing. And the second one, they couldn't save it, but they saved my face. So that's something that, facial paralysis is something that I think, it's something mentally, especially for somebody, who's in their 20s, they don't want to have facial paralysis, not obviously for the swallowing reasons and that kind of stuff, but, they just don't really want to have it in general for pictures or anything else. So, yeah.
Well, you still have hearing in one ear, correct?
I do. But I have a tumor on that side that is growing, but the Avastin has held it for 11 years off and on. So it's been, thank God. God.
That's really good news. Now back to Dr. Blakely. She mentioned Avastin, otherwise known as bevacizumab, monoclonal antibody. It's a big selling drug, commonly used for lots of different kinds of cancer, binds with VEGF. Can you talk about what the rationale was or is for this drug against this disease?
So, sure. The rationale now that we have data behind it, is that many of these peripheral nerve sheath tumors and also meningiomas are very vascular. They have a lot of new blood vessels, and that term is neovascularization. So even though they're benign, these are not cancers. They don't metastasize. They grow, and they grow by recruiting new blood vessels. And it turns out that many vestibular schwannomas have a very high rate of this neovascularization. And what we think that bevacizumab is doing is pruning those blood vessels down to control the rate of the growth of vestibular schwannomas, like McKinnon was saying. But maybe some other things, because one of the things that's really fascinating is that we can sometimes see that hearing gets dramatically better on bevacizumab, even though there's no change at all in the size of the tumor, or the tumor keeps growing, but the hearing is improved. So we don't totally understand that mechanism. We've done a lot of research on it in the last 11, 12 years, but when this option was presented to McKinnon, I knew about it because a colleague had offered it to a patient that he was taking care of truly on a whim. It is a drug that we were testing officially for a cancer called glioblastoma. It does benefit patients with glioblastoma, but we had no real idea of whether it would benefit patients with NF2 vestibular schwannomas. But in a person who really had no other options, he tried it. It worked really miraculously, thank goodness for that person. And then he tried it a few more times. Ultimately, that turned into a formal clinical trial. And we can now say that about 45% of patients with vestibular schwannomas due to NF2 will have both radiographic response in terms of shrinkage of their tumor and improvement in their hearing. And what's very different than in the cancer world is that for people who benefit, like McKinnon, it holds that effect for a long time, like more than a decade. Whereas in the cancer world, that's not true. We don't see the drug hold that effect on the tumor for so long. So we're very grateful for bevacizumab and its effect in NF2. You probably noticed I said 45% of people have benefit. That means that the other half do not have benefit. And so we've been on a pretty intense hunt to find drugs for the people who don't, the drugs and tumors that don't benefit from bevacizumab. But also bevacizumab takes its toll. That is, 10 years of protecting McKinnon's hearing, but at the cost of her going for infusions every 2 to 3 weeks, at the cost of skin breakdown, kidney damage. There are a lot of side effects that take their toll over time.
It's also an expensive drug too. But it is interesting. You mentioned that this came originally from a whim. Basically, it's one of those stories on how science kind of spills over from one area to the next, where it had been tested in glioblastoma, so people knew it could get into the brain, which was important. And this was theorized as a drug that would have to get into the brain too, and it's worth a try, even though there wasn't really animal data at that point to support the idea of that first person getting it.
Right. So we had no animal data, and I'll make a small comment that in the case of vestibular schwannomas and meningiomas, they don't have to get into the brain because they're not in the brain. They're on the linings just outside of the brain, and that's really important when we think about drug development, because the brain does have a specialized barrier that we struggle mightily against, but we actually don't have that barrier for vestibular schwannomas and meningiomas. We can get plenty of drug into those tumors. What's vexing is the pathways that are active in these schwannomas and meningiomas are normal pathways. And the cells, when you take them out, look like normal cells. So we have to find a way to treat the overactive cells without hurting the normal cells. And we have to find a drug that can differentiate those. And that's going to come down to finding a drug that can differentiate between the mutational status of the cells. So that's our challenge. It's not so much, unlike glioblastoma, we don't have to get it into the brain. We have to find the right target.
Are there any of these growth pathways that Dr. Korf mentioned that overlap with NF2 and give you some reason to think maybe one of those drugs that works there might work here?
Yep, that's why this is such a collaborative and communicative community. They all, these pathways are oversimplified when we draw them because we have but our own brains, but they are incredibly complex, and the pathway that is regulated by NF1, the RAS pathway, intersects with the pathway regulated by NF2 in at least 15 different spots. So what we don't understand yet is it tumor specific? Is it cell specific? Is it mutationally specific? That's the part we're trying to get to the bottom of.
So McKinnon, you've been on this bevacizumab for 11 years, I think you said. Have you been involved in any of these discussions about enrolling in other clinical trials, perhaps?
So I actually have been in another clinical trial, and it did not work. My kidneys were kind of taking a hit, and my right tumor was growing very aggressively. So we decided to switch before my second surgery, and I went into it, and it didn't work. But I mean, I think that's kind of like how we have to do this. Like, I mean, we're patients. Like, I want a cure, and like, I need to step up and go through these. Like, I believe in you guys, and you guys believe in me, and like, it's really a collaborative effort. So, I mean, that one didn't work, but the Avastin, the fact it did work just makes it worth it. So I'm excited about the future drugs that are coming out. I'm really, really excited about them. And I think that Avastin, we're continuing to stay on Avastin because it's working right now. And when it doesn't work, thank God right now, like I think I have some options that I didn't have before. So that's very exciting.
You mentioned the collaboration with Dr. Blakely. And I know, Rennie, you have this with Dr. Korf as well. Could you talk a little bit about the teamwork that you are bringing to bear here, and why that matters?
I think just because this condition is so rare, you really need the collaboration of a full team. Like, I think that with my medical stuff, like, I am constantly interacting with my doctors, and in turn, they constantly are there for me, which is amazing. And I think you really grow these relationships because it's my biggest anxiety is when I'm at the doctor's office, like those make or break moments, like we really share that together. So it's something that I really love my doctors, and I love all the researchers, and I love everybody that works behind this, because I know how much passion everybody has to put into it for us to all work together to be able to come up with these drugs, to be able to come up with a cure.
Same question to you, Dr. Blakely. Why is it important that you have that connection with the patient? Because, to do a trial, I mean, you have to make the case to your peers and talk to somebody at a drug company that might, have a molecule to give you. Where do the patients fit in?
Yeah, so the patients are the beginning, the middle, and the end of all of it. We wouldn't be motivated to be seeking these questions the answers to these questions in the first place if they weren't delineated to us so clearly. I mean, McKinnon's like one of my greatest teachers. She will say, this is what matters to me. You heard her say, my facial function is critical. That you must protect that at all costs. So that is a motivator to find a drug that will ensure that we stop growth of the tumors on the facial nerve. So that if we're not asking the patients what their biggest priority is first, you can waste a lot of years and money barking up the wrong tree. And so that's the first part. The second part is for all of the conditions we're going to talk about, most of the treatments will be required for a long period of time. And so we need to have that conversation with our patients. What's it worth to you? It's not me who's going for the infusions. It's not me who has to put up with this side effect or that side effect. So what is an acceptable side effect profile? And what do we have to deliver in terms of efficacy to justify that side effect profile? And then we can bring that back to our colleagues at the drug companies and say, I want a drug that I feel like that song, I want a new drug. I need a drug that does this, this, and this, and it's unacceptable if it does this, this, and this. And then we can sort through that. And you mix that data with the data that's coming out of our cellular experiments where we're choosing which drugs have the highest potency, and then what's coming out of the animal studies. And so when we choose these drugs, for example, the latest drug that McKinnon's talking about was discovered through a program called Encodes for NF2. My job at the table was to say, I'm never giving that drug to a patient. The toxicities are too extreme. We cannot have anybody on that drug for more than 2 months at a time. So next, what's the next best drug? And then we have the scientists telling us, well, this is why it should work. And the clinicians saying, well, this is what it needs to actually deliver to our patients. And the person who's telling me that, the people who are telling me that are people like McKinnon and our patients who tell us what the best drug should do.
It's great that you're open to that kind of feedback, because I'm willing to guess that when you talk to people at the FDA, for instance, they're not asking about the facial function preservation and what percentage of the population. They're just looking at other measurable.
I don't know. That might be the old FDA.
Okay.
The approval of selumetinib was at least a very high percentage of what they reviewed was the functional data, how patients felt, how their families reported they felt, and the response data on MRI, which is the traditional measure that we always think about in oncology, what showed the biologic effect, but the FDA was the one who said, show us it matters to patients. They cared very deeply about that. So I think that used to be the case. I think they are very tuned into, if we are not making life better for our patients, then they don't consider it effective.
I will like to add on to that. So I went down to the FDA to speak, and the care that the FDA showed towards patients, there was a patient panel, and the questions that they had, and you could see how much they really cared. So it was really nice seeing that. And I completely agree with Dr. Blakely. Like, that might have been the old FDA, but the new one, it was really, really amazing.
That's great to hear. I guess we would follow this under real world evidence and patient reported outcomes. These are kind of buzzwords now, but for a good reason. Okay, great. I think we should move next to our third, case study, if you will, on schwannomatosis. And Dr. Evans and Dale, maybe we could start again with Dr. Evans. Can you tell us a little bit about this disease, what it is, and how we have traditionally approached it?
So it's probably appropriate that schwannomatosis comes last. It's the late comer to the party. It probably was really only separated away from NF2 as a separate condition in 1997. And the first gene was only discovered in 2007 and the second gene in 2014. So it really is a late comer to the party. And the problem has been that really, as you hear in the name, schwannomatosis, the predominant tumor, by far the most common tumor, is the schwannoma, which is the predominant tumor in NF2. But the clear discrepancies are that you don't get ependymomas. Meningiomas are pretty rare, apart from in occasional SMART-B1 patients. And the predominant feature is pain from the tumors. That is the thing that really dominates the life of someone with schwannomatosis. And you can get rid of the pain by getting rid of the tumor, although not always. But pain is the overriding consideration. And where you heard lots of loss of function, because these tumors come out of the spinal cord on the nerves, they grow on the peripheral nerves. In NF2, they usually often cause loss of function, loss of muscular function particularly. That's pretty rare in schwannomatosis. In fact, usually it's the surgeon that actually messes it up in terms of causing the loss of function if the nerve needs to be cut to take the tumor off. So it is pain that is the overriding thing. Now, it's much rarer, prevalence of less than 1 in 100,000, although it's probably underdiagnosed. I think there are quite a lot of plastic surgeons out there who chop a couple of schwannomas off and the patient isn't told what they were, and they never find out that they've actually had 2 schwannomas, which technically potentially means they have schwannomatosis. You don't tend to get the tumors in the brain as much, pretty much not at all on the hearing and balance nerves with SMART-B1, and with LZTR1 only about 10% get it usually only on one side. So it is predominantly peripheral and spinal. And the other issue is that we've still only explained about 70% of the inherited cases. They only make up about 15% of all schwannomatosis cases that have inherited it from the parent. Only about 30% of the sporadic cases, so those with no family history, are explained. And then we go back to the overlap with NF2, because unfortunately in that group, if you don't find a fault in LZTR1 or SMART-B1, about half of those cases actually have mosaic NF2, not schwannomatosis. And you need to get 2 tumors out and look at both tumors genetically to prove that they don't have the same NF2 genetic fault. And that differentiates it from NF2.
How do you get the definitive diagnosis? Do you have to do like an exome sequence or something?
So, well, the definitive diagnosis is that you test for LZTR1 and SMART-B1, so you do a gene panel that tests all the main genes, NF2, LZTR1, SMART-B1. If you've got 2 schwannomas and you've got, or 1 schwannoma and you've got an LZTR1 or SMART-B1 pathogenic variant in the blood, then that's a diagnosis of either LZTR1 schwannomatosis or SMART-B1 schwannomatosis. If both of those are absent, no genetic fault, then you actually, to formally make a diagnosis, you need to get genetic material from 2 schwannomas to be certain, if there is no affected parent, that you're dealing with another maybe gene on chromosome 22 yet to be discovered, or even a gene not on chromosome 22, but to show, because the predominant, pretty much in every schwannoma, you lose both copies of the NF2 gene, that the genetic faults in the tumor are different between the 2 tumors, not the same. If they're the same, even if they're not present in blood, you're effectively saying this is mosaic or partial NF2, where the genetic fault has developed not in the egg or sperm that made that person, but sometime during development as an embryo.
This is one where the tumor heterogeneity can be really confounding.
Yeah, absolutely. And it's a much more complex diagnosis than NF2 and NF1, really, because you can usually make those diagnoses confidently clinically, although not always, but usually you can. Whereas with schwannomatosis, really to definitively say it is, although the pain and the tumor distribution usually gives you a strong clue, but you still need to have a genetic diagnosis to be sure.
Okay, so the pain is really the big symptom that patients are coming in and they need
overriding symptom.
And they are, how are they getting treated for that? I mean, I can imagine, you know, if they don't have a good diagnosis, they get
all sorts
type
and
things.
most things, yeah, all sorts. You know, start off with paracetamol and you go on to anti-inflammatories, and they are next to useless at treating schwannoma pain. And really, you know, you usually end up on opiates at some stage, so morphine derivatives. But you know, that's pretty bad thing for your quality of life and your ability to do normal things and work. So the predominant drugs are drugs like pregabalin and gabapentin, which even those aren't brilliant for your work, but those are drugs that are good for neuropathic pain. But even those often don't work in people with schwannomatosis. And so we're crying out for a drug that is targeted at what is causing the pain in schwannomatosis.
Okay, thanks for that explanation, Dr. Evans. Dale, can you tell us about your story, how you came to learn that you had this disease?
Yeah, my story started out about 2004. I started noticing some lower back pain. By about 2005, early 2005, my friend was sort of sick and tired of hearing me, you know, complain about it. So he said, "Why don't you go get an MRI?" I have a couple friends that are spinal surgeons, and both those surgeons came back with the same diagnosis, saying a degenerative disc disease. You're getting older. It's a sort of part of growing up. So I took that sort of as an answer. I was given some physical therapy, which I didn't feel like was doing much, and life progressed over 10 years, and being sort of a sandwich with some aging parents, some newborn children, as my pain increased to the point where, you know, you touch my back or touch, you know, my knee, you could drop me, you know, as a sort of a sack of potatoes on the floor. Yeah, I went in for a routine physical and had my jacket on, was going to walk out and saying goodbye to the doctor. And he said, "How's your back pain?" And I said, "You know, it's just incredibly painful." And he says, "Well, come back in. Let's take a little closer look." And he said, "That lump should not be there." So that led to an MRI. 4 tumors were found in my sort of sacrum area. Referring surgeon sort of had a general idea of neurofibromatosis. And I sort of remember writing it down and sort of, he says, "We'll do some biopsies," and it came back as a schwannoma. And he referred me to a Parkinson's clinic about a week later. And I didn't really get past the receptionist call because I sort of called in disbelief. And at that point, I sort of did a simple Google search and found, you know, that living in Minnesota here, there's an NF clinic at the University of Minnesota and Mayo Clinic. I had appointments at both, got into Mayo first. They put me through the system very quickly, determined that I had schwannomatosis with some of the information that had been revealed from MRI. I had surgery scheduled, you know, about a month later, and had two tumors removed at that time, and that was just a fabulous relief and an improvement in quality of life. But as you know, I read some, you know, information. CTF website was great. I realized that, hey, spinal tumors are common here, so I asked for a spinal scan, and you know, within about a month of my surgery, and that revealed tumors there. And about a year later, I was like, guys, I'm got this leg pain that, you know, I'm wondering if this could be a tumor. And sure enough, a MRI there led to a revelation that there were a couple more tumors down there in my leg. So, you know, you sort of, you know, you get to the point where you're sort of, you know, living life and thinking that your body's just sort of getting older. And now every once in a while you sort of think, oh, could that be another schwannoma growing? And so you got that sort of hanging in the back of your brain every once in a while. But doctor's been great. You know, this group here has been awesome, and you know, getting the information out there so that you can sort of be your own advocate, you know, to you know, chase that diagnosis. Something I missed. I took the first diagnosis as the correct diagnosis, and I missed 10 years of getting maybe treatment sooner.
But once you got your diagnosis and then you had that surgery, that sounds like it was successful. What about those other subsequent tumors that were discovered? What did you do about those?
I had one more surgery, so removed 2, and then the other ones are sort of monitoring. So it's 6-month scans and comparing, you know, growth to prior, and sort of charting that out. So it's yeah, scans every 6 months.
Okay, now back to Dr. Evans. Hearing what your colleagues have talked about with NF and, you know, it's related, what are some of the best ideas out there for, you know, new treatments for schwannomatosis?
Well, I guess we've got to start really with bevacizumab Avastin, because that has been tried on a few patients. We've treated a couple with great success. That particularly it seems to be pretty good at the pain, and we've learned that as well from some of our NF2 patients who have particularly a big spinal tumor load with pain, that it shrinks the tumors, and it's really quite good at treating the pain. Expensive painkiller, obviously, but I think what I think that tells us is that what works in NF2 is likely to work. If it works on NF2 schwannomas, it's likely to work on schwannomatosis schwannomas, because again, the predominant driving event in the tumor is loss of the NF2 gene, loss of both functioning copies, no myelin protein from the NF2 gene, functioning protein to work. That causes the cells to proliferate and grow and form a tumor. If you can get into that pathway and stop that happening, and we still don't know fully, as Jayshree says, how bevacizumab works completely, and it's sort of in a peripheral way, particularly with the VEGF to the what's predominant in the NF2 pathway. But I think, you know, what we can learn from NF2 is almost certainly going to tell us what's going to work in schwannomatosis in terms of controlling and shrinking the tumors. But where, you know, NF2 and schwannomatosis are a bit different is that pain is not a predominant feature. It does occur, it's not a predominant feature, but it is the predominant feature in schwannomatosis. So if we can find a way of better treating the pain than we currently can, and it is very imperfect in schwannomatosis patients, that is the way forward. But as Dale has told you, surgery can be incredibly effective at treating the pain. And the thing about schwannomatosis is you can get pain with a 2 or 3 millimeter tumor that's very hard to pick up on an MRI scan. You actually have to really do dedicated close MRIs of the, we've dug out little tiny intercostal nerve between the ribs in order to get rid of the pain. It can be really, really odd how just such a tiny tumor can cause so much pain. And it's probably secretory in some way that's causing the issue.
Interesting. So now, since this is an even more rare population, I would imagine certain kinds of hurdles present with, running a rigorous clinical trial. Number one, with recruiting enough patients. Do you run into that?
Absolutely. Well, we haven't been in a situation yet. I mean, Scott Plotkin, who is talking as part of this event, has set up a clinical trial for a pain medication, and we just weren't able to get that up and running. I mean, we do have quite a lot of patients in the UK, because we've been pretty good at getting the diagnosis on the map in the UK, and so I think we would have enough patients to get a trial going. But a lot of the patients actually have had all their tumors removed and don't have a problem at the moment, and so you wouldn't actually have someone that would be useful in a clinical trial unless you're looking purely at preventing tumors growing in the future, which is a much more expensive trial because you have to wait a long time to see whether it works. So yes, it is a small pool, and it's even smaller because you have less patients with actively growing tumors that are easy to monitor.
That's a hard trial to run for practical reasons, but in all likelihood, there probably are some residual schwannomas, if they're as tiny as you say, and can have that kind of effect.
I mean, that's the whole thing, that you think someone presents with schwannomatosis, and they often do, with very segmental disease, and they've got just loads of schwannomas down one arm, But when you do a whole body scan, you find other schwannomas dotted around the body. But for some reason, and this may well be genetic and something that's happened in embryogenesis, it predominates in one region. That maybe another genetic event has happened in that area during development as an embryo, which makes that area particularly susceptible, like maybe the sacrum, if that's where Dale has had his predominant tumor load.
Okay, we only have a few minutes left, so I want to ask, and this could go to anyone, I think actually the patients first. How do you think about the role of patient advocacy groups and the internet in terms of like facilitating better communication, coordination of research and care?
Well, the first place people go to is the internet, regardless of what their physician tells them as soon as they walk out. So it is what it is. And there's a lot of really scary information on the internet, like McKinnon alluded to earlier. So I think one of the strongest benefits of patient advocacy organizations is putting accurate information out there that's based on science. And, the physicians that are on this webinar today are very active in terms of the advocacy organizations and making sure that we have the accurate information to be able to be our own advocates and to be realistic about what the future may hold for ourselves or our loved ones. I think it's also very important for the advocacy organizations to advocate for the drug development like we have had and what's been alluded to on this call. And I think both Dale and McKinnon and I, some of these drugs were not available when we were, our children or ourselves were diagnosed. And now the landscape is really wide open, and we're very hopeful that, like selumetinib, that other drugs, are ramping up. So whereas what felt like we were going 5 miles per hour, I'm hopeful that now we're 200 miles per hour and that this is going to open doors for other treatments for other people and other conditions associated with the neurofibromatosis.
McKinnon or Dale, would you like to comment on how the internet and the patient advocacy groups have been helpful?
Yeah. So obviously, kind of as Rini said, like, you go straight to the internet. And I think that's something to keep in mind, that just because it has all these things, I mean, the internet is not completely inaccurate, but just, it doesn't mean that like that all is going to happen to you. So there's so many variations, I feel like, of NF2 that doesn't mean that it all happens to you. So it's good that you get to know like the information yourself and be your own advocate. Like, there's so many times, like I had said previously, that like 10, 11 years ago, a lot of doctors didn't even know what NF2 was. And so everywhere I go, everywhere I go, even like if I'm like in line talking to somebody, I always try to educate them, because I think it's so important for us to continue to educate as many people as we can, because how do they know how to fix something if they don't even know what that is? So I think patient advocacy is so important, to speak up for yourselves, because the doctors speak up for you. Like, why can't we speak up for ourselves also?
Great point. Dale?
One thing I learned is, a rare disease is not all that rare. 1 out of 10 have a rare disease. So I think, as McKinnon's saying, when you're out there having a cup of coffee, standing in line, you have an opportunity to educate on NF, as well as learn about somebody else's rare disease. So I think, the internet, this sort of allows, us to become better informed, people, neighbors, friends, and help those within not only the NF community, but those, next door that may not have NF, but another rare disease.
Dr. Blakely, I saw you kind of smiling and nodding early in this. Like, a lot of doctors I talk to, they maybe roll their eyes a little bit. The patient comes in and they got their diagnosis from Dr. Google. It's not that simple, right? But how do you try to make it actually useful for your interactions with patients?
So there's no denying that we all go to the internet. And in fact, as McKinnon was telling you, oftentimes patients will tell us stories that they go to see a physician who doesn't know about NF1 or NF2 or schwannomatosis, and they watch them Googling the term while they're in there being evaluated. So it's a real thing. I do think having trusted patient advocacy groups like the Children's Tumor Foundation that vet the information that gets posted on that website, not only with the experts in the field who are doing the research or coming up with the diagnostic criteria, but also with patients, So it's vetted from both populations before it's posted. At least I do on my sheets can tell you, I say, these are the websites that I endorse you looking at because this has accurate information. And then the next part is that no, you're not on any website. So what's going to happen to you or what is happening to you isn't on those websites. That we are figuring out that story together. So you can find out what's happened to somebody else, and maybe that will inform an important conversation so that we can address whatever, those anxieties are so real and we have to address them because otherwise they just build and grow their own lives. But it doesn't predict what's going to happen to that person.
Well, as the journalist in here, I would add that the patient advocates not only can provide accurate information, but they can put it in plain English so that it's not in the Journal of Neurofibromatosis, and that the patients can have a productive conversation with their doctor.
There actually was a journal called Neurofibromatosis.
I was just making that up.
There was one though.
Which 50 people read. Yes.
And another thing, the many NF advocacy groups, the CTF does this, and other groups in Europe do this. They point patients to the specialty clinics. And so it's not easy to get to a specialty clinic all the time, but it's at least you can know who those people are. And most specialty clinics are not in it to make things hard for patients. We're happy to pick up the phone and have a conversation and see if we can find a path for that patient. And I do think maybe the single greatest thing any patient can do for their own care is have a consultation at least once with a clinic that specializes in their condition.
One other thing to keep in mind here is that most of the time when we're talking about instituting a treatment for any of the forms of NF, whether it's surgical or medical, it's a shared decision-making process. There are occasions, like if your spinal cord is compressed, maybe you have no choice but to do surgery. But the vast majority of the time, there needs to be a really in-depth conversation about the risks and benefits of any particular approach to treatment. And that's a shared process between the physician and the patient. It's not a one-sided, here's what you absolutely have to do, and it's not your choice here. The more informed the patient is, the more meaningful that conversation can be.
Well, that's great. I think we are out of time. So thank you all so much for a very informative, interesting conversation about NF, and really a hopeful one too.
Thank you.
Thank you.
Thank you.
Thank you, Luke, and thank you all speakers for enlightening discussion. As you highlighted, in the fight against the deliberating diseases like NF, collaborations cannot be limited to scientists alone. We are all in this together. Thank you all for the sharing. So now let's move to the second track today. Let's hear from the movers and shakers behind the making of the first NF1 breakthrough therapy and the pipelines under development for NF2 patients. Hosting the session today is Rich Saul, head of the Boston office of WuXi AppTec, as well as its strategic initiative advisor. So Rich, the floor is all yours.
Good morning. Thank you for joining us for the second of today's two webinars from WuXi AppTec. My name is Rich Saul from WuXi AppTec, and I would like to welcome all of you. This webinar discussion will feature two examples of non-traditional drug discovery and development that demonstrate the true potential of effective and transformational collaborations. We'll talk about two drugs, Koseluego and brigatinib. Now, Koseluego is the branded name for a drug known as selumetinib. It is the first drug to be approved for the treatment of NF1. Koseluego achieved several milestones along its development path. It was designated as a orphan drug designation in 2018, rare pediatric disease designation, breakthrough therapy designation, and finally approval from the United States Food and Drug Administration on April 10th, 2020 for the treatment of NF1. The second drug is brigatinib. It's an approved treatment for the lung cancer. It's currently in the beginning parts of its clinical trial development for the treatment of NF2 by a mechanism that is distinct from its use against lung cancer. Non-traditional drug development, particularly for rare diseases, is a growing specialty in the healthcare and research community. In today's environment, There's both a growing investment in and appreciation for the importance of collaborative networks in successfully delivering meaningful and transformative medicines to our patients. Often these networks involve a number of key stakeholders that would include clinicians, entrepreneurs, biotech and pharmaceutical companies, researchers, regulators, capital investment groups, patient advocacy groups, as well as the patients themselves. We're honored to be joined by a distinguished panel of advocates, medical professionals, and scientists who've graciously agreed to share their insights and experiences in this timely discussion. With me today are Annette Backer, President of the Children's Tumor Foundation, Brigitte Weideman, Chief of Pediatric Oncology Branch and Deputy Director for the Center for Cancer Research at the National Cancer Institute, George Kirk, who's the Global Development Lead for Carcillugo and Adavosertib at AstraZeneca, Michael Humphreys, the U.S. Scientific Director for Lung Cancer at Takeda Oncology, and Scott Plotkin, Professor of Neurology at Harvard Medical School, Executive Director for the Center for Neuro-Oncology and Director at Mass General Hospital, Dana-Farber Cancer Institute, and Brigham and Women's Hospital's Neuro-Oncology Fellowship Program. Please join me in welcoming them. So I'd like to address my first question to Brigitte, and maybe you could say in a few words, talk about NF1. How is selumetinib's successful development differ from the traditional drug discovery, and how this drug came to your division for development, and what you found in your clinical trials?
Thank you so much for inviting me to talk about this story. Neurofibromatosis type 1 is a fairly common genetic disorder caused by a mutation in the NF1 tumor suppressor, NF, and about 1 in 3,000 people develop NF, which causes a myriad of tumor and non-tumor manifestations, many of which are progressive, and for none of them an effective approved medical therapy existed. I'm a pediatric oncologist with a field of NF1 for tumors called plexiform neurofibromas, which develop in half of the patients or people with NF1, many of them in very young patients. These tumors are not cancer tumors, but grow progressively and can cause serious and life-threatening problems. As an oncologist, I was interested in identifying active therapies for NF1, but there were many hurdles that we had to overcome, which I'm happy to talk about. But after overcoming all of these hurdles, we actually evaluated at the NCI Pediatric Oncology Branch and other sites selumetinib, or Carcillugo now, as a medicine to target these tumors in young children. And to our great, we were very happy, but also surprised to see a response in the very first patient. And then we saw it in the second patient and in the third patient after many negative clinical trials. We saw tumor reduction, which gave us hope that this might be beneficial for patients. We partnered with CTAP, the Cancer Therapy Evaluation Program of the NCI, and AstraZeneca, and George Kirk is here to talk about this today, and developed a registration study into many, many others invested a lot. This was a true NF community effort. This trial completed enrollment of 50 patients within just 1 year, and we were able to show a very challenging task, not only that we shrink tumors, but that patients benefit from the treatment by feeling better, having less pain, and having a better quality of life. And this then ultimately led to approval of selumetinib, or now Carcillugo, for these patients. It's a pediatric drug approval. It's very different from what we typically see, where adults come first. And we're very excited. It's a very long story, and many, many have contributed, including the people here. The CTF that built the NF community provided funding for certain aspects of this. Dr. Plotkin, who's the leader of the RAINS International Collaboration, the Response Evaluation in Neurofibromatosis and Schwannomatosis Working Group, when he in 2011 said, let's develop something that we have standardized endpoints for clinical trials, which was very much embraced by the Food and Drug Administration. The AstraZeneca team, which has been phenomenal to work with. It's been a lot of work, and I have a much better appreciation now what goes into getting a drug on the market, by just following a little bit of what George and his team have done. There were many others, but first and foremost, I want to mention the patients that went through multiple evaluations on the registration trial. not because it was important for them, but for us to show that the drug actually works.
Thank you, Brigitte. George, AstraZeneca has pretty broad familiarity with selumetinib. How do you, from your perspective, how is the drug development of this drug non-traditional?
Yeah, thanks, Richard. It's a good question, and I'll build on quite a few things that Dr. Wiedemann said as well. I think there were three main aspects from my perspective that made the development of Koselugo non-traditional. The first one, which Dr. Wiedemann has touched on, is the collaborative effort that was required. I mean, we do collaborate a lot with academic institutions when we're developing our drugs, but in the case of this development in NF1 specifically, and as Dr. Wiedemann mentioned, we actually collaborated with a number of groups to design and deliver the registration study in these patients. And that included CTEP, as Dr. Wiedemann mentioned, but as well as Dr. Wiedemann's group, NTAP, and the CTF, and more importantly, the NF1 community. And it's hard to explain how important that collaboration actually was to the success of the trial. So that's the first aspect. The second aspect, which is non-traditional, is the Koselugo is the first drug that's been approved in NF1. So it's really quite historic, but the challenge that gave the team was that there was no regulatory precedence for an approval in this indication. So for the regulators, and in our case the FDA, this was new to them as well. So actually working closely with the FDA over the last 3 years has been really important. And we took Dr. Wiedemann with us to the FDA, we took CTEP along with us as well. So we really went there as a collaborative team. And it's fair to say we worked with the FDA over the last 3 years to try and make this happen. And fortunately, we have been able to do that. The third aspect, which I wanted to highlight, which is non-traditional, although it may sound traditional, is the patients. And we all talk about, across all the pharma companies, about how the patients are really at the heart of everything that we're trying to achieve. But the example we have from the registration trial in NF1, plexiform neurofibromas, the patient stories were really pivotal to the regulatory review and to the regulatory approval. Being able to describe the clinical benefit these patients received when their tumours or their tumour volumes were reduced by treatment with Koselugo was really important and pivotal to help the FDA understand what that clinical benefit meant to those patients. And we included those patient stories in the regulatory submission. That is definitely not traditional. We're very used to looking at our normal statistical analysis of these studies, which we did as well. But the patient stories played a big part in the approval for Koselugo and NF1 plexiform neurofibromas. So I think those are the three main aspects I would highlight as being non-traditional, Richard.
Thank you very much, George. Scott, NF2 is a basically distinct disease. Would you like to comment on that, please?
Thank you very much. Thank you for the invitation to be here. So neurofibromatosis type 2 is one of the three conditions that we lump together under the term of neurofibromatosis. NF2 is less common than NF type 1. It's about 1/10 as common, about 1 in 25,000 births. It's a condition that's characterized by the predisposition to develop multiple benign tumors, including schwannomas, meningiomas, and spinal ependymomas. In particular, the schwannomas are located in the vestibular nerves, or the 8th cranial nerves. This is in contrast to NF1, which is characterized by the predisposition to form neurofibromas, which are a closely related but distinct type of nerve sheath tumor. So while they share a common predisposition to nerve sheath tumors, they are different types of nerve sheath tumors with different anatomic localizations. In addition, the presentation of NF2 typically occurs later in the late teens or early 20s, in contrast to NF1, which is typically in the first decade. There have been many historical precedents of lumping them together, but now through genetics, we know that these are two distinct conditions that will require different approaches to develop therapies.
Thank you very much, Scott. So, Annette, the discovery of brigatinib was really discovered by a completely different mechanism, or approach, I should say, that's what I'll call wholly non-traditional. Can you elaborate on that, please?
Sure. Thank you, Rich, for inviting me to this very distinguished panel. I'm very excited to be here. Yes, it was a very different approach. So the approach we took with NF2 was, in fact, we listened to the patients. So it's like what George says is that the patients in the NF community are very savvy and they play a very different role than in certain other communities. The patients came to us and they said, we are reading all these papers about NF2 drugs that are available. And some papers say it works and some people say it doesn't work. And for them, it was very confusing. So what we did is we said, okay, we as a patient advocacy group are going to throw this question out to the world. And then we also said we're going to launch, I always say, a micro stand-up to cancer initiative where we really want global experts to come together to fix this very unsolvable problem, or let's say at first seems unsolvable problem. So the team came together. There were 4 co-PIs, which Scott Plotkin was one, Jeffrey Blakely, Wade Clapp, and Jim Gazella. They came together and they said, not only do we want to develop a preclinical pipeline that will deliver new drugs for clinical development, we also want to understand why certain drugs are working and why certain drugs are not working. So they have, in fact, over the course of about 4 years, built a unique preclinical pipeline that has led to the discovery of brigatinib. And then there was an extra player that in fact came in later in the course, which was Encodes, which is the National Center for Advancing Translational Science, because in fact the early testing of the drugs didn't really deliver a drug that would work in all the cell lines and in all the models. So Encodes offered their screening capabilities to discover potentially new drugs that nobody had thought of. And brigatinib was in fact that molecule. And the mechanism that brigatinib is working in NF2 tumors is not the mechanism for which it was approved in lung cancer. That publication is submitted, so I would be careful disclosing the mechanism, but it is definitely a different mechanism, a mechanism that is relevant for NF2, and that is why it was so unexpected, because the approved mechanism was not the mechanism that is acting in NF2.
Thank you. So you took the data and you went to Takeda to help support the development. So to Michael, how did Takeda support that development in NF2, and how is it different from other Takeda programs?
Sure, I'm happy to answer that question. As everybody else mentioned, I'm really happy to be here today. Really appreciate the invite. So as you heard from my title and from the indication that brigatinib is approved in, my specialty is lung cancer. And we feel very comfortable in terms of evaluating brigatinib in lung cancer related programs, whether that's preclinical or clinical studies. It's currently approved in a later line setting for ALK-positive non-small cell lung cancer. And we currently also have a package that's going to the FDA for a broader indication of this drug. And again, we feel very comfortable with this, but science and data emerges and evolves as you move forward. This occurs in the small space we work in lung cancer. This also occurs more broadly in oncology in general and other disease states as well. So it's just a classic situation of we don't know what we don't know, in that we have a program that allows us to, for people to submit unsolicited requests for different types of trials and bring that data to us so we can evaluate it very rigorously and work in a collaborative fashion with these investigators to see if it's something that would make sense to move forward. And this fell in line with our strategy, and I'm really happy to be able to support it moving forward.
And how are you supplying the drug? So you're supplying the drug, right? To the
so we're
trials.
Supplying the funding for the trial. This is what's called an investigator-sponsored trial. We supply the funding, and we utilize the expertise of Dr. Plotkin and his team to run the trial.
And along those lines, Scott, you were the PI on the Intuit 2 trial. Now, this is a broad investigation of the drug within NF2 and And do you see this as a more effective or efficient design than a typical trial in this space?
Yes. Thank you for those questions. I would like, before I explain some of the comments you brought up, I would like to explain one of the unique things about the way that the Children's Foundation organized the Synodos preclinical pipeline was that they insisted that there would be clinicians involved in this research along with the preclinical experts. And for that reason, I and Jayshree Blakely were embedded within this preclinical pipeline program for 5 years before we got to a lead compound. And that enabled us to work very effectively with Takeda. And they have been absolutely spectacular partners. I can't say enough about that collaboration and the willingness to think outside the box in terms of developing drugs for rare diseases. So when we had a lead compound that was showing broad activity in multiple preclinical models, that is preclinical models of schwannomas, which are nerve sheath tumors, as well as meningiomas, we wanted to design a trial that would be unique and, more importantly, efficient. So we took some of the novel concepts in clinical oncology, including a platform trial as well as a basket trial, and applied them to NF2. In the case of a basket trial, these trials often will lump together in a basket, in different baskets, different tumor types that share common genetic alterations. So you might have ALK mutant lung cancer and neuroblastoma. That might be 2 baskets of a trial. In our case, we wanted to lump in different tumor types driven by NF2 loss. In this case, that would be schwannomas, meningiomas, and ependymomas, and make eligible patients with progressive tumors of any tumor type. This is the first time we've had broad eligibility for a clinical trial, and we were very focused on equity here to try to include as many patients as possible for some of these promising new agents. The second key aspect of the design was a platform trial. And in a platform trial, you establish common eligibility criteria as well as common endpoints so that you can incorporate a design that has multiple drugs over time. So for example, all the patients come into the platform that we've called Intuit NF2. The first drug that will be studied on the platform is brigatinib, as I mentioned, but we anticipate, hopefully with further collaboration with Takeda, who've been very open to discussions, additional drugs that have been identified in the preclinical pipeline entering this platform trial, such that patients who unfortunately maybe don't have a good clinical response to brigatinib could then return to the platform and then be re-randomized to a new drug. And the idea is, unfortunately today, if you're setting up trials serially, that is one after the other for patients, it could take years to get a patient on a new trial. In this design, we hope that will shorten that time from years to months and also provide value to Takeda and other companies by rapidly having patients go between the different arms. The final thing is, because they have common eligibility criteria, it provides something beneficial for rare disease, and that is comparison between arms. Not direct comparison that provides statistical strength, but really a comparison that these are homogeneous groups that allows us to at least understand a little bit about these different drugs. And it's a very unusual design for rare disease, and we were so pleased that Takeda was willing to go on this journey with us. Back to you, Rich.
Wonderful. And maybe one quick thing to add, Rich, is that so this trial design has also, now we have in Europe a European grant that is called EU Pearl, which is also looking at platform trials for all forms of NF. And we just had a consensus meeting less than 10 days ago, where in fact the agreement was that the Intuit trial would definitely be an example trial to also reach, let's say, launch you in the European community. So it looks like we are globalizing a lot of our efforts through this collaboration.
Yeah, it's wonderful. Scott, how long will what's the duration of the trial, and how many folks are you looking at to enroll?
Well, again, the way I think about it, we have a platform portion, and we hope that that'll go for about 10 years. And then there's an individual study arm. And in this case, that would be the first trial of brigatinib. We hope that we will be able to fully accrue that within 2 to 3 years. And I'll tell you, sometimes we've done better than that, even though it's a rare condition. We have wonderful partners that are all coming together. And so we hope to cycle through multiple drugs during this 10-year platform trial.
Okay, thank you very much for the update on that. So in the course of identifying and working up these compounds, clearly there were a lot of collaborations, and we've gone through them. And how important were they to your efforts? Let's start with Annette.
Yeah, I think what we have been looking at CTF is, in fact, instead of being a funder, we have been looking at what is that unique role that we as a foundation can play in the NF ecosystem. And when George, for example, said also the interactions with the regulators, we had patients that were, in fact, in front of the regulators to explain to them what it actually means to live with NF. We had 5 or 6 patients that went to the FDA and spoke for an hour and a half, in fact, to the regulators. We also see ourselves as interacting with the pharmaceutical companies, like with George, like with Mike. There are these interactions with pharma companies that just make it easier, and we can really help. And we see ourselves as that building that R&D non-for-profit enabling platform, if you want, to really make sure that we can not only catalyze new discoveries, but that we actually also not end with the publication, but that we actually, as soon as discoveries are made, that we help translate those discoveries into clinical benefit. And we do that by offering platforms such as the registry, the clinic network, et cetera, et cetera. And to come back, for example, to the MEC story, to the selumetinib koselugo story, the interesting thing is, and we published a paper on that, is that was really a funding community come together. So CTF had seed funded some discovery work, but then, for example, the NIH and CDMRP, which is a congressionally directed medical research program, they really have invested a lot of money in understanding the mechanistics behind the MEC mechanism in plexiform neurofibromas and other tumors. Then CTF has invested about $11 million over 6 years or 8 years in the NF Preclinical Consortium, which really demonstrated that if you inhibit MEC, you indeed shrink tumors. We helped fund the first in human, which again, a seed funding. And then in fact, the big funding came again out of the NIH NCI. So it is really, every funder has played a unique role in making sure that we have this kind of successes that we're announcing. And for brigatinib, it's the same situation, right? We helped fund and we managed this Synodos team. Then Scott and Jayshree had managed the interactions with Takeda, and now we're co-funding with Takeda the clinical trials. So it's kind of, what is that unique gap that a non-for-profit can play in bringing not only discoveries to the table, but also make sure that those discoveries actually benefit the patients.
That's wonderful. Scott, would you like to add into that, please?
Yes, I agree, of course, with everything Annette said. I'll go back to a previous comment to highlight the incredible role of our preclinical colleagues that played, at least in the NF2 discoveries, and really to thank them for their collaboration, because, as I said, the clinicians were embedded in this effort. And as we watched the process, we were able to add very critical, I think, advice about this drug is closer in human trials and might be more reasonable. This drug, while it's interesting, may not be. And that's because we knew from the beginning and agreed at the beginning that the end result in this case was going to be a drug to enter human studies. It was not going to be just a mechanism based, and that's not to say that that's insufficient, but in this case, it was very directed. And I think knowing where you want to be when you finish was incredibly helpful. And our preclinical colleagues really supported that effort, and that's very special, I think.
And Michael, could you describe your interactions with the various folks along the way in terms of the collaborative environment?
Yeah, I'm happy to do that. And I'm a preclinical scientist by training. George, I'm not sure you're aware of this, but I work for Array Biopharma, which initially developed selumetinib. So I have a long history in preclinical science, and I have to say, brigatinib is a selective ALK inhibitor. It also inhibits another protein called ROS1, quite similar potency to ALK. but to a lesser degree, it inhibits other kinase targets as well. And those could be clinically meaningful, and I think that's what we're hoping plays out here. There's a few targets that roll up into that profile. And I have to say that the preclinical data that I saw was fascinating, and it was robust, and I believe that was really what formed the basis behind us being interested in moving forward with this trial.
So you interacted very closely with the Children's Tumor Foundation, I guess, and poring over the preclinical data, yes?
Sure. I mean, to be completely candid about it, I've done it on the outskirts. Some of my colleagues have been much more involved in it, but I have seen some of that, yes.
Okay. So, Brigitte, you had a lot of interesting collaborations along the way that I only came to appreciate in the course of prepping up for this interview. And so how did you come across the drug to begin with?
Yeah, so I thought I wanted to echo a little bit what everybody just said, is that, I go back to your question too, that we all do a unique part. And when I came to NF, it was through the annual meeting that we have that is sponsored by the CTF, where I, as a pediatric oncologist, learned about NF and got fascinated and hooked. Meet the basic researchers, the clinical researchers. Basic researchers thought it was crazy what we were doing in the beginning, but I think we've become so much closer in the collaboration. Drug development is what I do, so I do want to quickly highlight, I know I'm not yet answering your question, but I did come to this because these NF tumors are driven by upregulating the RAS pathway, which is very much upregulated in human cancer. So we thought this is going to be easy to deal with, and we developed a clinical trial that was sponsored by the Cancer Therapy Evaluation Program of a farnesyltransferase inhibitor. I know I networked with those agents in the past too. It didn't work, and then CTEP sponsored another trial with sorafenib, and it didn't work. We worked at the NCI on a natural history study to develop methods how to measure these tumors, how to follow them, and how to understand the development of morbidity, which was very cost intensive. And at some point my boss said, shall we stop this study? And I was very glad later on to hear that the FDA really wanted to see this data. But this is what led to the MEK inhibitor. We thought, okay, tipifarnib didn't work, sorafenib didn't work. There was some preclinical data when we started the trial, and CTEP took a huge leap to say, yes, let's do it, because we had already 2 negative studies, and the NCI also said, yes, let's do it. And then really wonderfully, the preclinical consortium produced beautiful data showing, as Annette showed, that selumetinib and another MEK inhibitor worked consistently in these mice, and nothing else had really worked. So that was a huge point of validation to move forward. This is how I got into this, and I do want to mention another really important aspect of this collaboration, which was once we saw that something worked, that we really needed to have a partner to work with, and that was AstraZeneca, but also insight from the community and from experts like Scott's group and the Rains group. We went to the FDA actually for informal discussions to educate them and learn from them about NF and how we could develop endpoints. That has been a very, very strong collaboration and needed. And I almost feel the FDA is a collaborator in this entire area because they have been so insightful and helpful in helping us move along.
Is there anyone else would like to add into that by any chance?
Rich, there is one. There are 2 things that I would like to add. Is first of all, I completely agree with Brigitte that working with the FDA these days is fabulous. They are collaborators. Before, when I was still in J&J, they were more like the judges. You would give them your packet and they would say thumbs up or thumbs down. Now I really feel, especially in the rare disease community, we have an amazing collaboration with the FDA. And we just, as an example, we had a gene therapy webinar this morning at the foundation, and the director for biologics was one of the panelists. So the FDA is really part of the exercise, and they want drugs for patients. The second, I think, very important element is the role of the patients in the NF community is, I think, very unique. And maybe, Scott, you want to speak a little bit about the RAINS initiative. Our patients are really partners in the whole R&D process. they evaluate our clinics, they evaluate our grants, they participate in endpoint development. I mean, they're very different people than the traditional patient who is waiting for the treatment to come. But I don't know, Scott, if you want to add something.
Yeah, the thing I'd like to add, I think that's exactly right. I would emphasize, though, that I do think that other communities can use this as a blueprint, and I hope we'll talk about this in the next stage. Although NF patients are wonderful, it's one of the reasons that I've been in this field for more than 20 years. It is, I think, a blueprint for other conditions. And what I would say about the RANGE group, this is an international voluntary collaboration of physicians, scientists, patient representatives. I do include the FDA, CTEP, and any other group as well as pharma that's involved. It's anyone can come. The goal is to develop drugs to help people. That patient representatives have been integrated since about 2017 and are equal voting members in the committee. And as Annette alluded to, they are tasked with providing their unique perspective in these discussions. We know that investigators and clinicians have a very unique type of knowledge built of study and science, but patients have a very unique personal lived experience that physicians and others don't have. And as the FDA in the US stresses patient benefit as part, to make somebody's function, life better or live longer, that really helped sharpen our focus on endpoints that became the backbone of some of the work with the MEC program that Dr. Wiedemann has been talking about. So I think it was critical. I think it keeps us honest as investigators. And I think that it's definitely doable. And I know that we're publishing a lot of our expertise in this specifically to see if other groups are interested in following kind of the blueprint that's worked for our community.
Right, and we'll come back to the blueprint in just a moment. So George, you've had a lot of, I guess AZ has lots of experiences in collaboration. How is the Costa Lugo experience been for you?
Yeah, thanks, Richard. And before I answer that question, I just wanted to say I agree with everything that everyone's saying so far. But how did it work different? How was it better for us? I think a lot of it comes down to the people, Richard. I think working with Dr. Wiedemann and her team, they made it very easy to collaborate. And that's down to the strength of the science in that team, the technical aspects and their knowledge and experience, but also just their willingness to be open with AstraZeneca when we're talking about data that they had, their ideas, and also willing to listen to our ideas as well. And I think that's what worked really well from our perspective compared to maybe some other collaborations. But I know we're going to come on to this because Scott mentioned it as well. But this really is a blueprint for collaborations in the future, because the other aspect I wanted to highlight of going back to your question, why was it important to have this collaboration? For me, the two things I wanted to say. One, without this collaboration, we would not have Koselugo approved in the US. It's as simple as that. If we didn't have the CTF helping and shaping the preclinical science, if we didn't have Dr. Wiedemann's team, taking that leap of faith with CTEP to start the phase 1 study, we would not be where we are. And from an AZ perspective, we can only thank the CTF, CTEP, and the NCI for really driving that forward. But I think the second aspect in terms of why this collaboration is so important, and to answer your question as well, working with Dr. Wiedemann and our team, and also Dr. Jessry Blakely from NTAP, we became almost like one team. We were not separate entities, if you like. We were separate organizations. And an example of that is the approach we took to the interactions we had with the FDA, which, as Dr. Wiedemann has and our team have learned, which was new to them, it became much more numerous as we got closer to the regulatory submission. And Dr. Wiedemann and our team were with us the whole way, the whole journey. We didn't do that ourselves. We did that as one team. It wasn't just AstraZeneca taking the submission to the FDA. And that for me was quite unique and really important to the success of the collaboration.
I have some questions from the audience that has come in. I'd like to pose this one to Brigitte about what was the most challenging obstacle to designing the right protocol for the clinic with a MEK inhibitor?
It's a great question. When we did the phase 1 study, we took a very traditional approach. We said, we'll define the best dose, and we studied the pharmacokinetics. And then we saw so many patients respond. And I remember distinctly the FDA in a meeting we had with George saying, when we thought of breakthrough therapy designation, he said, well, you have to show us the benefit. You lost an opportunity in your phase 1 study. And then we sat down, and that's where Rains also came in and thought, okay, tumors can be in the eye, they can push on the airway, they can be at bowel and bladder. How do we document the benefit? We needed a set of different endpoints and outcome measures that we would look at clinically in each and every patient, in addition to showing that there's shrinkage. This was really a formidable task, and I was always a little bit afraid that we might fail showing the benefit because the tumors are so variable and some are established for a long time. Will we really see benefit after tumors have been there for several years? I have no doubt that Koselugo or selumetinib works for patients, but was this going to be the effective strategy? And so with the FDA and AstraZeneca and the Rains group, the plan was to have the volume reduction as the primary endpoint because we can measure it and we have shown we can do this consistently. And then because there are no validated outcome measures yet for the pediatric population, all of the other measures were key secondary endpoints. And we were glad to show that we could show improvement in pain, disfigurement, quality of life. And George mentioned a really interesting aspect. For every single patient enrolled, we had a vignette where we showed for each individual patient what changed. And the investigators provided a judgment if they thought it had benefited the patient or not. It's very non-traditional. So it was really everybody putting their head together and thinking, how can we maximize this? And this is where the thanks to the patients come in. Some patients would be admitted for a sleep study. That's an overnight admission, pulmonary function test, a walk test, range of motion, strength, vision, you name it, depending on how many morbidities they had. So it was very challenging, but I'm very glad that this worked out, and our strategy appears to have been quite successful.
Selumetinib and brigatinib are both kinase inhibitors, and it's not very often you find those kinase inhibitors outside of the cancer setting, and think about indications that it should go in. So I would think it would have a very important to be very safe, particularly since these are being used in a pediatric setting. Could you comment on that?
Oh yeah, I can. So I think I'm a pediatric oncologist. So for me, it was clear we had to start in pediatrics because we see kids ending up with an artificial breathing tube at age 2 or 3 with these tumors. So industry had to, and CTEP buy in, this is something we have to look at in kids because these tumors grow the most rapidly. But then we actually started another clinical trial after we saw our successes in pediatric patients. And we thought, oh, tumors in adults don't grow quickly, but adults have a lot of clinical problems with these tumors. And so we actually did start another CTEP-sponsored study in adult patients. We only have preliminary data, but it looks quite promising for adult patients as well. And I do think the adult patients deserve just as much, obviously, attention. And so we're very eager to explore what the benefits of Koselugo may be in adult patients. And I don't know if George wants to comment on that or has any additional thought.
Yeah, thanks. So the only, to answer your question, Richard, in terms of the safety profile, the majority of the adverse events we saw in the trial that Dr. Wiedemann and our group ran were of grade 3 or less. So in our minds and in our opinion, manageable in terms of adverse events and a manageable safety profile. And the majority of those events actually resolved during the course of treatment, which is good news. And I think the other thing to point out with Koselugo, it has been studied in a large number of adult patients in cancer trials, other tumor types, as you mentioned. Actually, the way and the dose in the pediatric patients is less than the dose that we tend to give in the adult population. We still see the efficacy, obviously, and it also helps with the safety profile as well.
Thank you. George, there's a question here about global drug development. Can you comment on that? for the drug? Yeah.
Sure, yeah. I mean, as we mentioned at the top of the call, Koselugo was approved in the US in April for the treatment of NF1 plexiform neurofibromas. We have submissions, other regulatory submissions ongoing in other countries, in particular in Europe. And we're also in discussions with regulatory authorities in China and in Japan for submissions in those countries. Both of those countries require us to do a study in patients from Japan and from China. So we are planning to start a phase 1, phase 2 study in Chinese patients, hopefully in the second half of this year, all being well. So there are plans to submit in other countries outside of the US, because our intention in AstraZeneca, now we have the positive data and the approval in the US, is to try and make Koselugo available to as many patients as possible who have NF1 and plexiform neurofibromas.
Well, thanks very much for that update, George. I'd like to come back to Scott for a moment and talk about the blueprint idea that he brought up. The question we have so far, the question we might ask to ourselves is, what have we learned so far in doing this kind of R&D and development in the non-traditional way? What have we learned? Is this blueprint that you're talking about going to be scalable? Scott, why don't you take a look at that? Thank you.
I think that this model is very scalable, but I'll emphasize that it's not easy. I do think that this began in the 1990s, I think, or maybe even 1986, when the Children's Tumor Foundation, then called the National Neurofibromatosis Foundation, initiated an annual conference that brought together the top scientists around the world. And that community, which is a warm, inclusive community, has grown over time, and I think really forms the backbone of everything you just heard about, whether it's the RAINS initiative or these collaborations between the clinicians. And I think really underpins some of this willingness of the investigators to work with CTEP and drug companies. I think it's really a characteristic of our community, of which I'm very personally proud. I think that there's a couple key points to emphasize that need to be duplicated, in my opinion. One of them I'm going to ask Dr. Wiedemann to talk about when she takes over in a second. That's the importance of natural history studies for rare diseases, and nobody has done this better than the pediatric oncology branch at the NCI. And so I'm going to defer that to her, because that is critical for rare diseases to understand the natural history. I think the other part is the RAINS group working on endpoints. Again, in rare diseases, you tend to have to make your own endpoints and validate them. And partnering with the FDA, with pharma and other groups, I think is critical and can be done. We have found it very easy to do those collaborations. And then finally, to find the right partners in pharma. And I would say my experience has been great across the board. Takeda has been wonderful, but many other companies have been as well. I think you have to be patient, and it's up to you to bring these other tools to bear, your natural history studies, your collaborations, and help them understand why their compounds and why their resources will be well used, in these efforts. So I think there's a lot to learn. I know that everyone in our community is happy to help any other community that's interested in this as well. Brigitte, can I ask you to speak about natural history studies, because I think it's so important?
Sure, and I think I want to also highlight how much we try to learn from other communities. There's others that we can obviously learn a lot from. The NCI Natural History Study started in 2008, when we really did not have a good understanding of how these tumors grow. It's a resource-intensive study. As I mentioned, when I talked to Dr. Bruce Korf, an eminent NF researcher about it, he said, "This is my dream study." And I said, "Why don't you participate?" He said, "Well, this would be prohibitive at his institution." And I think I alluded to the fact that at some point, even my former chief said, "For much longer do you want to do this?" But it was really important, and the FDA asked for it, that we not only show that we benefit the patients, but that Koselugo, in this case, really alters the natural history of the disease. And I was so happy that we had this data, and AstraZeneca really worked hard with us to make sure we could analyze the data in the optimal way. And what we essentially had is years of growth of these tumors in children of the same age than the patients that enrolled on the trial, and we could document that their tumors grow for the most part and never shrink beyond maybe 1 or 2% or something like that. And that was in stark contrast to what we saw with selumetinib. So I very much agree with Scott. Thoughtfully designed natural history studies, and they may not need that many resources, but this is going to be critical for any rare disease. And the FDA recently issued guidance for industry that anybody can look at for natural history studies. This is one of the things I think that we can do uniquely at the NCI intramural program, where we are not held by grant cycles. So I'm not being told I'm a failure if I don't produce something within a short period of time, and being able to collect this data, I think did make a meaningful contribution.
Scott, this platform has interesting potential, particularly in rare diseases where it might be difficult to run particular kinds of studies in a traditional way.
Yes, I do. I think that there's some common themes here for rare diseases. It's very hard to do what I would call traditional oncology studies, where you do a phase 1 with 10 to 100 people, and a phase 2, 50 to 500, and a phase 3 with hundreds to thousands. That avenue is not realistic from a patient perspective, much less a financial perspective. And so we need to use techniques like natural history studies, well-designed studies with clear endpoints, and I would suggest some novel trial designs, like we have hopefully in the platform basket study, that'll make us more efficient. And so I think that kind of flexible thinking is available, and drug companies are willing to go down that path with you if you can show them why that it makes sense and will be hopefully productive at the end. So for me, I think it's easy to get bogged down when you're working with rare diseases that you can't get a drug approved because all you see is how a drug gets approved, you know, in a very common cancer like breast cancer. And I would say that's not true. Many of these ideas are very applicable to other conditions. You just need some experts to help bring the ideas together, I think, with a group that's dedicated to pushing it forward.
Yeah. Annette, would you like to comment?
Yeah, I've always looked at collaboration as a value proposition. I always feel that it's not a question of collaborate to collaborate. The value of together should be more than the value of apart. And that is, I think, something that is really important. And I think there is a couple of characteristics that I have learned to appreciate during the years, because it's true, collaborate is not always easy. If I would say the 4 things that to me collaboration should be, if not, it's not working, is trust. There needs to be a vision, there needs to be passion, and there needs to be an element of fun. And if these 4 elements are in the mix somehow, collaborations are successful. And then there is one absolute essential element as well, is the donors. If we would not have the funding, we would not be able to do what we do. So I think that's also a very important aspect of the mix. So to have the people on the one hand, and I think that is the strength of the NF community. I always look, if I go to the conference or if I look at the meetings, there is really not just the head, but there is also a heart factor. People want to do good. People want to bring better treatments to their patients. And I think that is also what has made this community unique. It may sound cheesy, but it is really important to keep motivating and to keep being enthusiastic about what you're doing. What the blueprint for me would be, if a community can have those elements, then I think, yes, this is absolutely a blueprint. But there has to be a will.
So Michael, how would Takeda's view on this obviously is through the brigatinib must be very positive. And what about it, more broad strokes than that? Well, Shire, you have Shire already. For rare disease.
Was the question in terms of collaborations? I mean, go ahead.
Well, a blueprint. So how would the big pharma, fit into this at the end of the day? Is just going to be a broader mechanism for development, or do you see this partnership as being very, very critical, the collaborative partnership between, let's say, yeah, go on.
I think Takeda likes to believe that it is a patient-first company, and we're constantly looking for innovative therapies, evolving science, and methods to drive science and medicine forward. Whether that's related to our primary indication or our primary driver behind a drug. Obviously, for brigatinib, it's ALK-positive non-small cell lung cancer. That is the most extensive patients that have these specific types of molecular alterations. But as we talked about, brigatinib has other activities as well. So we're certainly interested in exploring those and doing everything we can to provide value to patients and have strong collaborations with our folks in the medical field and the physicians that we work with.
Hey, George, any thoughts from AstraZeneca on a blueprint model?
I mean, I think this can be applied to all rare diseases. I agree with Mike. I think Takeda, AstraZeneca have very similar philosophies here because the science was at the forefront of what we did on the NF1-PN studies, and for NF2, science is driving the way forward there. And I think companies who have that philosophy to be open to scientific innovation, that's where we will succeed. But applying this to other rare diseases, it can definitely happen. But the only comment I would make, and I'm not trying to be negative, it is challenging, but it's very rewarding at the same time. So go into it with your eyes open. There's a challenge, but if you put the right kind of positive energy, if you like, into it, and you hold your nerve. That's the phrase that we used in AstraZeneca for this. We really held the nerve because we knew at the end of it, patients were getting benefit. That was the most important thing to us. And even if you're working in a rare disease, if you know that there are patients at the end of it who are receiving benefit, if you keep that in your mind all the way through, then you can make it successful.
Well, thank you very much. So we're pretty close to the end. I think we'd like to go into some closing remarks. Maybe I'll start off with Brigitte.
Yeah, no, I enjoyed this panel very much. We hope that there will be many other reasons to have panels like this again, to celebrate other successes. And I mostly really want to thank everybody who was involved in this. So many, I'm sure many that we did not mention. It really is a large group. And I think everybody, including every single patient who participated in any clinical trial directed at plexiform neurofibromas, get some credit to what has happened. Thank you.
Thanks again for allowing me to participate today. I actually learned a lot by participating in this today from these experts here, and really appreciate the chance. And we're really looking forward to seeing the brigatinib arm of Dr. Plotkin's trial open and begin accruing and hopefully providing some value to some patients out there.
Yeah, again, I want to thank you for inviting me to this panel. It is a true honor to work for the NF patients, and it is also a true honor to be in company with so many smart people that have a passion to end NF. So I really wanted to thank everybody. And you too, as an extremely active board member.
Yeah, thanks, Richard, and thanks for the invitation for taking part in this panel today. It's been really, really interesting, and I always enjoy working with all the people on this panel, so appreciate the invite. I just wanted to, end by saying a big thank you to the patients and the families in the NF1 community, because without them, none of us would actually be sitting here telling this story.
Thank you for pointing that out. I, of course, echo everything that people have said in closing, the gratitude for our patients and for our colleagues. My own comment in closing would be, there has never been a more exciting time to be in rare diseases. I cannot tell you how interesting and exciting and promising it is now, with the number of molecules coming out, with the preclinical screening that we can do, with the blueprints for collaborations that we have. So if you are a junior person listening to the webinar, it's time to jump in with both feet. If you're somebody more seasoned like myself, hopefully you'll enjoy the ride. And for me, the special sauce is the real pleasure of collaborating with other people, patients, investigators, foundations, and so forth. And I think this panel is very illustrative of that. So thank you for allowing me to participate.
Oh, thank you all. I'd just like to close this webinar by saying, clearly collaborations play a very important role in creating transformative drugs. And we've seen this already in the case of selumetinib, and certainly we look forward to seeing positive results coming out of the brigatinib trials and the future drugs to come out of Scott's platform as well. So thank you so much, and we hope that this model that we're talking about will be very broadly applicable to other rare diseases. And thank you all for your time here. Thank you. Thank you all.
Thank you, Rich, and thank you, speakers, for your great perspectives. As you highlighted, now more than ever, creative collaborations are critically needed in order for us to accelerate life-changing solutions for patients like Rayleigh, McKinnon, and Dale. As we close today's program, we would like to encourage you to join the many activities this May around the world, hosted by leading organizations like Children's Tumor Foundation. Together, we can help raise awareness and help spread the spirit of collaboration. Thank you everyone for joining us today. We look forward to seeing you at our next episode of Collaborations That Transform.